Now it's time to actually get to our notes, and we're on page 77. So if you'll go to page 77 in your notes, what we need to start by talking about is what really is evolution. And so we want to make sure that you know that when we're studying evolution in science, even though the word means change over time, like cars change over time, and fashion changes over time, that's not exactly what it means when we're studying how life changes over time. Now, when we study evolution in science, it's really easy to just look at a bunch of facts and say, well, that's just a theory. What we're going to do in class is we're going to look at case studies and real data, and we're going to see how do you best explain that data. And we are going to have to go back in time, and we don't have a time machine, so we'll be looking at fossil records, and we'll be looking at evidence that shows that organisms change, living things change, but they don't change over time. You can't change. Your genes are already set, but over generations. So your offspring might be different from you, and their offspring might be different, and that's where evolution really comes in. We're going to go way back to where did life start?
So, with the origin of life, first we start with abiogenesis. This is the idea that life came from non-living material, and that's also called spontaneous generation. In the early atmosphere, so when Earth first started, all the gases that were in the air were mainly carbon monoxide, nitrogen, and carbon dioxide. Big thing, there was no oxygen present.
So when we look at this theory, we can actually break the word down into, we should recognize bio. And when we recognize bio, and I'm going to change the color and make it a little bit bigger if I can. Bio, we know, means life. But A means not. And you might know that Genesis means beginning. So abiogenesis literally means that on early Earth, there wasn't life. We can actually trace fossil records back and see a time where there were no cells. And this also makes sense if we look at the other planets. There are planets with no life on them. So abiogenesis is the idea that life, cells, might have come from non-living things. How does that make sense? We actually already know that non-living things might be things like molecules. And that makes sense. We know that cells are made of molecules like phospholipids and carbohydrates and proteins and nucleic acids. So this theory is supported by fossil records that show that there was a time where there was no life, and in fact the atmosphere was even really different. And so abiogenesis is the idea of how life started. So it's the theory of how life might have started. There are lots of different theories, but this one is kind of supported by an experiment that we'll see next.
So then we had Operin, and he proposed the primordial soup hypothesis. His idea was that the first molecules that were present could have been organic molecules due to the early conditions of Earth. And so because of the carbon monoxide, carbon dioxide, and nitrogen,
we're adding this energy, and so we'll see it in the next experiment. But this is kind of the first hypothesis, that we had inorganic molecules. Some examples of inorganic molecules. We see them right here, carbon monoxide, carbon dioxide, and nitrogen. If we remember what inorganic means there is no CH and O, and organic means there is CH and O. And we actually have our 4 organic molecules that we know. So the theory is, where did these molecules come from? And that's what we'll look at with the next experiment that actually supports the primordial soup hypothesis.
So Miller and Urey were 2 scientists that tested his idea, and so they created the early Earth conditions in the lab, and they actually observed that organic molecules could form from the molecules that were there.
The big thing that we have to add here is that early Earth conditions actually were very unstable. So if you can add that into your notes, if you'll add that early Earth was unstable. There were lots of eruptions and lightning, and the earth is moving. Remember Pangea and how the earth's crust was moving, and there's lots of earthquakes. And that energy is what they tried to get in this experiment. If we have water, and that's H2O, if we have carbon dioxide, and we add a lot of extra energy to it, we could actually form organic compounds. And so that was Operan's theory, and they tested it in a lab, and they actually got organic molecules. Now keep in mind, that's a really long way away from getting a cell. Getting a protein is not the same as getting a cell. So the question is, will we ever be able to see life made in a lab experiment? No. This happened over 1 billion years. The first cells weren't seen until 1 billion years after Earth had existed. So we're never going to see this in a lab, because you can't watch a lab experiment for 1 billion years. And so even though we have evidence to support, because this supports abiogenesis, does it show us a cell? No. But it gives us evidence that we could make the parts to build a cell. And scientists are still working on this, but it's not like we're trying to make life come out of nowhere. This is just how life started. How you could have had an empty planet, and how all the conditions on that planet might have allowed life to first start. But that's not what happens now.
So with biogenesis, this means that life must come from life. So life began from abiogenesis and continues through reproduction.
And so it kind of is confusing. It makes you think, well, abiogenesis means life from non-living. Biogenesis means life from living. Aren't those opposites? Don't they contradict each other? Abiogenesis is how life started. Biogenesis is how life continues now. So Earth is not the same as it was 4 billion years ago. We don't have an empty planet with lots of unstable conditions and no oxygen. Now, life is not going to just burst out of molecules. Now we see that life comes from other life. So cell division, mitosis, meiosis, go back to sexual and asexual reproduction. So it's not that they contradict, but that they actually work together. Abiogenesis was first, and biogenesis explains how life exists now.
So then we had Francesco Redi. He disproved abiogenesis, which occurred. He disproved abiogenesis occurring now with his rotting meat experiment. So he proved that maggots came from flies and not from rotting meat.
So this is an interesting experiment because meat doesn't really rot much for us because we have refrigerators. But before, so this is in like the 1500s, you didn't have a refrigerator. And so meat just kind of sat out, and all of a sudden it would be rotten and there'd be maggots on the meat. And people would wonder, where did the maggots come from? And it turns out that the maggots actually came from flies landing on the meat and laying eggs. Maggots are actually kind of like caterpillars. Think of a caterpillar and a butterfly. Maggots are just the larvae form of the fly. And so what he did is he covered the jars. He left this one open, he covered this one with paper, and this one with gauze. The one that had paper, the flies didn't get on. There were never any maggots. The one that was open, that the flies got on, flies landed, maggots grew. The one that had gauze, air could still get in, flies could not. No eggs, no maggots. And so people used to wonder, where did life come from? They'd look at a pond and be like, well, where did that fish come from? And so Redi was able to prove that life is really coming from other living things.
Next we had Louis Pasteur, and he disproved abiogenesis as well in an experiment where he had single-celled organisms in his S-shaped flask.
And so for a while people were like, okay, so a fly can't burst into life. A fly comes from another living thing. But what about something really small? Could a single cell just come to be? And Pasteur did this experiment where he actually took chicken broth, which has bacteria in it. He boiled it, right? We know that boiling it means you add heat, you denature the enzymes. And so boiling it should kill anything. And then he left it out open. Now this S shape allowed air to get in, but because of this gravity pulling down, no bacteria could ever get in, up, and then back down. These flasks have been sitting there for over 300 years, and nothing has ever grown in them. But if you break the top off the flask and you let what's in the air, bacteria and fungus, fall in, then you do actually get contamination and bacteria growing. So it's this idea that abiogenesis may have happened 4 billion years ago, but it's not happening now.
So with the evolution of cells, we have the heterotroph hypothesis. In this hypothesis, we had our first cells were prokaryotic and anaerobic. And this is because we had no oxygen in the atmosphere. So our heterotrophs would actually consume food.
So think back to the primordial soup. The primordial soup had molecules that the first cells could have consumed. So if you form a barrier, if we happen to have some lipids that form a barrier and we have a cell, really all that a cell is an inside and an outside. So if the inside is able to then consume food around it and grow and change, then all of a sudden we have a cell. They were going to be prokaryotic because they're still simple, and they're anaerobic because there is no oxygen, and that's really important. So we're not saying that this was one first cell. This probably happened multiple places and times over Earth's early history. But then there were some changes, some mutations.
So then when we get to our second cell, our cells were still prokaryotic, they were anaerobic as well, but then they became photosynthetic. So this means that there was some oxygen being produced.
And so the key here is we're still pretty simple, and we're still mostly anaerobic, but there was a mutation. And now these cells could make food. If we think about what photosynthesis means, plants, think of a plant, plants make food and produce oxygen. Well, this is not a plant, this is still a single cell, but it has the enzymes to take carbon dioxide and water and build its own food. Now keep in mind that we're not saying the first cell evolved into the second cell. We're talking about many generations, so reproduction, 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 accumulating mutations. And so we're still going to have some like this, but now we also have variety or variation.
Then with our third cell type, now we've become aerobic and prokaryotic cells. The atmosphere is now filled with oxygen due to those photosynthesizing prokaryotic cells.
So we start at the beginning with prokaryotes. Anaerobic, heterotrophic. And then we see some that are still prokaryotic and anaerobic, but they might be heterotrophic, they might be photosynthetic. The third example was prokaryotic, but now I might be aerobic or anaerobic. I might be photosynthetic, I might be heterotrophic. So we see this huge variety of prokaryotic cells, but that doesn't explain all the cells on Earth.
So then we have the evolution of our eukaryotic cells, and this is explained through the endosymbiont theory. This explains the evolution of our eukaryotic cells.
We're going to go ahead, and you might have to pause the video to actually get this written down in your notes. So if you need to, pause.
So with the